Hi grade 10 learners, welcome to Learn Extra. Today we're going to be looking at transport and support systems in plants. Let's go look at some of the key concepts we'll be looking at today. In this session, part 1, we'll be looking at summarizing what you need to know about the anatomy of dicots. This will include the root and stem system, how the cells and the different tissues are distributed. Then the structures of these different cells in the different tissues, because as you know, each different structure of the plant will have a different function, and their cells are adapted to help them with this function. Then we'll also be looking at secondary growth, how the annual rings of a tree trunk in a tree can tell you of what that tree went through in the previous years, droughts, fires, things like that. Then we'll be looking at how the tissues in a stem are specialized for their different functions. So let's go right to it. So let's start with the dicot root. If you take a look at the following diagram, on this side here, you'll see it's the external structure of the root. And what we did with this one is we cut right through the root, and you can see if you look at it from the inside, and this is what we'll be looking on later on, is the internal structure. So let's now look at the external structure as it's here. So first of all, you see that we have the root cap right here at the bottom. Now this root cap is slimy and it protects the root because, as you remember, the root needs to burrow into the soil. It's very hard, and this root cap and its sliminess helps the root cap to penetrate the soil, and it protects it from getting hurt. Right behind the root cap here you get the meristematic region. Right there. This is the meristematic region. And what happens here is this is where cells continually divide by mitosis. So this is where your root grows. This is where new cells come and they divide and the root elongates. Behind that, you will get the area of elongation. What happens in the area of elongation is that the cells grow larger, and this is where your root also elongates. Then you have your root hair region right here. You have thousands of tiny little root hairs, and the root hairs is there to absorb water from the soil. It enlarges the surface area and it helps to absorb water and minerals from the soil. Then that is your external structure of your root. Now if we go and look so we said that we have the root cap right there, and I said that it protects the tip of the root and it is slimy to facilitate movement. Then under the root cap you have the meristematic region, and this is where cells divide. This is where you produce new cells by mitosis. Then you enlarge in size in the region of elongation. This results in the root growing, and then the root hair region to absorb water and dissolve mineral salts. And then the root grows wider and may produce lateral roots in the mature region, which is just behind that. Now if we go and look at the internal structure of the cell, of the root, if we look, this is now where we cut the roots laterally, and we're looking at it from the top, and this is what you'll basically see. First of all, you're going to get your root hair. This is formed out of the cells on the epidermis, which is that layer of cells right there. Then you get your cortex. After your cortex, you get your endodermis, which also has the Caspian strips in them. We're going to look at the functions of each of these just now. Behind your endodermis, you get your xylem and your phloem and the pericycle. And then the parenchyma cells as well, that makes part of the cortex. So let's go look at the functions of each of these cells. First of all, there's no waterproof cuticle that surrounds the root because the whole purpose of the root is to absorb water and minerals. So if you have this waterproof cuticle, that function would not be possible. Later on, when we go and look at the stems, you will see that we do have a waterproof cuticle there because we want to prevent water loss. Then the epidermis. Your epidermis, as we said, it's the single layer of cells that goes around the inner tissues, as you can see there. Here's your epidermis, the single layer of cells, and it goes around the rest of the tissues, and that is for protection. And out of there, as I said, you also get the root hairs. Then the next thing is your cortex, and in the cortex you get your parenchyma cells. Now these parenchyma cells are very large because it's there that you store your waters and minerals. Then you get your endodermis. Your endodermis contains the Caspian strips, and this facilitates movement of water through the endodermis. Then you get your stele. And the stele consists of the pericycle, the xylem, and the phloem. Now your pericycle, if we go back to the diagram here. So this is your stele, right there. And it consists of the pericycle. Now it is from your pericycle that your lateral roots will form. Then you get your xylem, and your xylem is responsible for transporting waters and minerals that the root absorbed and taking it to the stem and the leaves. And then you get your phloem, and the phloem is responsible to transport the food that was made via photosynthesis from the leaves, and it takes it down to the root. So this is basically what your internal structure of the root looks like. Now let's look at the transport or the movement of water from the soil into the root to the stems. Now this diagram shows it very nicely. So basically what happens is this is your remember this is now your root right there, your root hair that grows from the epidermis, and then this is your soil, little soil particles. Now what happens is the water that is in between the soil particles moves into your root hair. Water moves, you remember the concept of osmosis, where water moves from a higher concentration to a lower concentration, and the water moves from the soil where it's a high concentration into the root hair, and then it goes through the epidermis, through your parenchyma cells, right into your xylem. So I mentioned that this is the pathway of water through the soil, through the root, right into the stem. Basically what happens is you've got your water in between your ground particles. It moves via osmosis into the root hair, into the root hair's vacuole. Through the root hair it goes across the epidermis and into the parenchyma cells. Now from here there's two ways that the water can move into the rest of the root. The first way is that it can either go by osmosis into the vacuole of the parenchyma cells and move through the parenchyma cells across the cambium strip into the xylem and into the rest of the plant. Or it can just simply go through in between the parenchyma cells. So it doesn't move into the parenchyma cell itself, it just moves in between it into the xylem and into the rest of the plant. And that's basically it for roots. We're going to take a short break now. I need some caffeine. So right after that, we're going to be looking at the stems and the internal structure and external structure and things related to that. Welcome back, grade 10 learners. I hope you had a nice break. Just before the ad break, we were looking at the root structure of the dicot root and stem structure, or the root structure, and we look at the internal and the external structure, and we took a look at the different cells and how they are made and how they help with the structure. Now we're going to be jumping to the stem of the dicot. Sorry. I'm sorry. Next time I'll make my hair into a ponytail.
From the top?
Yes, we can start from the top. I'm ready.
5, 4, 3, 2, 1.
Welcome back, grade 10 learners. I hope you had a nice break. Before the break, we took a look at the dicot root system and how the structures and the cells are specialized to help with the root and its function. Now we're going to be looking at the stem of the dicot and how, again, the internal and the external structure and how the cells are specialized to help with the structure, to help with the function of the stem. So let's first look at the external structure of the stem of the dicot. Now, if we just quickly look at this, you'll see that I'm going to draw you a little diagram here. If I can I make it bigger? Let's make it green. So you will have your dicot. Okay, I'm not a graphic artist, but. So now if I say that you have your nodes, basically this would be a node. Internode, I'm sorry. So you would have a node there, a node there. This would be your internode. In your node, you will find your auxiliary buds, and this is where your lateral stems will form from. And at the end of your, okay, let's just go back. So when I say leaves develop from nodes, so there you find your nodes. The spaces between the nodes are called your internode. So this is your space between the nodes, and this is called your internode. Then in the node or at the node, you will find an auxiliary bud. And the auxiliary bud is where the side branches are formed from. Then at the end of the stem, right at the top, at the end of the stem, you will find the terminal bud. And the terminal bud is what allows the stem to increase in length. Now this is just basically your external structure of the stem. It's very straightforward. Let's go look at the internal structure of the stem. This diagram is a cross-section and it shows the internal structure of a young dicot stem. So basically what we have here, it's very similar to the dicot root, same terms and everything, so you should be familiar with that already. So if we start and we look right on the outside, we have the cuticle, which is right on the outside, even outside of the epidermis. Then underneath that we have the epidermis. Then we have your Casparian strip, your endodermis right there, the pericycle, your phloem, your cambium, your xylem, and your parenchyma. So let's go see how this all fits together so that it makes sense. So right on the outside, if you remembered, we said that you have a waterproof cuticle. And this is even on the outside of the epidermis, and this is to prevent water loss. Remember when we discussed the root, we said that there is no waterproof cuticle in the root, as the function of the root is to absorb water. Now on your stem you do have a waterproof cuticle because you want to prevent water loss. Then you have your epidermis, and your epidermis has a single layer of cells, and it is again to protect the underlying tissues. So if we take it this, we said there's your cuticle right there, and then you have your epidermis, which is a single layer of cells, and it is to protect all these tissues on the inside. Then after your epidermis, you have your cortex. And your cortex is made of parenchyma cells. Remember, parenchyma cells are those large cells, and they basically store the water and the minerals that your plant absorbs. So it's your cortex and your parenchyma cells that stores water and food. If you look at the diagram, I'll quickly show you where that is. We only have the parenchyma. Here's all your parenchyma. Your cortex, I'll show you later on in another diagram exactly where the cortex fits in when we look at a whole cross section. This is just a tiny bit of a young stem, but when I'll show you in the whole cross section of a more then. Okay. Then you have your vascular bundles, and your vascular bundles are arranged and ring around the medulla. If I can quickly draw you, they will basically look like this. If you have your cross section of your stem there, your vascular bundles are going to be arranged like this. And then in there, Each of the vascular bundles are going to consist of a pericycle, which contains the sclerochema cells. The sclerochema cells, you remember from the previous lessons, it is basically dead cells, but they are very strong, so they are for structure and support. That's what keeps your plant upright mostly. Then you're going to have your cambium. In your cambium, you have your meristematic cells. Your meristematic cells are the ones that continually divide by mitosis, and that's what causes the plant to grow, or in the case of a stem, to thicken. We're going to be looking at that in your ear ring section. Then your phloem. Phloem is the same function as in the root. It basically transports the food that your plant has made via photosynthesis from the leaves, and it takes it downwards to your roots. And then you have your xylem, and your xylem basically transports your water from the roots where it's absorbed, and it takes it up to the stem. So you basically have this two-way highway of xylem and phloem that you're working with. So if we again go back to that, you have your pericycle, and then you said you have your phloem. Your phloem is there, and then you have your cambium, and then you have your xylem. Now, if I can explain to you, this is now a cross-section of a young plant. Remember where I drew you the diagram of how the stem would look in the cross-section, if I can do it again here. Now this part is basically, if we took this vascular bundles and you took, if I can do it like this, it would be, so that is basically that part that we have a diagram of. So if I take one of these vascular bundles, and I can show you where you get your, and I just draw it here, it will be like this. You will first have your endodermis, which will be here on the outside, the endodermis here. Then you will get your pericycle. This is the pericycle then there. And then you will get your phloem, your cambium, your xylem. So you will have your, this will be your cambium, your phloem will be here, your xylem will be there. Now I said we're going to be looking at secondary growth. If you look at a plant, and we always say on our plant a tree, and you cut it a cross section, you always see tree rings. It makes very nice patterns. And how these tree rings are formed is what we're going to be looking at now. So basically we say here that every growing season, the stem of a plant increases in width. It basically becomes thicker, and this is called secondary thickening. So what happens is, towards the end of the first year of growth, the parenchyma cells between the vascular bundles. Now remember, where is this? Let's just reorganize ourselves. Where is the parenchyma cells between the vascular bundles? So this is all parenchyma cells. This is your vascular bundles. And these are all parenchyma cells in between there. All this is parenchyma cells. You see there, xylem parenchyma. It's all these cells is parenchyma. And they become meristematic. So these between the vascular bundles just, so these ones will become meristematic. They become meristematic. And then you have your cambium strip. Remember, there's your cambium strip. So these cells become meristematic, and they join up with the cambium strip there. So this whole region there becomes meristematic. So you can already see where this ring is going to form. Back. So the parenchyma cells between the vascular bundles, they become meristematic, and they link up with the cambium, and then they form a cambium ring. They form a cambium ring. So now this cambium ring starts dividing. Remember, you have your phloem on the outside, your xylem on the inside, and in between them you have your cambium ring. Now these cells have become meristematic. They start dividing. The phloem divides to the outside, the xylem divides to the inside, and now you have your stem increasing in width. So let's look at it like this. So basically, here is your bark. Then you have your cambium, your core cambium, then your secondary phloem, your vascular cambium, and this is the things where it links up with the parenchyma cells and it becomes meristematic. And then you have your xylem. So the growth happens in this direction and in this direction, because both your xylem and your phloem becomes meristematic, and it starts dividing in both directions. It's not the xylem and the phloem that divides, but the meristematic cells here at the vascular cambium. They start dividing towards that direction, and they start dividing towards this direction. So the cells in the cambium ring start dividing, and they form secondary phloem, and we said on the outside, and then secondary xylem on the inside. Each year you get another ring of secondary phloem and a secondary xylem that is formed, and this makes the stem wider. And that is how your growth forms. Your plant increases in width. Now basically when we talk about tree rings, and this is a very nice picture of it, the tree rings you can see, and all that it is, as we said here, it is a layer of wood, and it is produced during one tree's growing season. But this can tell us a lot about the tree. Now when we look at this one, this is your bark on the outside, and you can see your tree rings. Now this one here is the first year of growth. Now when you look at it, this is a rainy season. Basically you can see this one in relation to this one. You see how broad this one is? It's very broad. So there was a lot of rain. The tree could really grow a lot. This one was a dry season. It's very narrow. So you can see there wasn't a lot of water available. The tree couldn't grow a lot. This is a scar from a forest fire. So then you can see during that year there was a forest fire. So let's go look at what they say now. So we have a cross section, which is what the diagram in the previous slide was about. And now it's concentric tree rings. So each year as it grows, the tree forms another ring. And you can see, you can first count the years, how old the tree is, and then you can also tell a lot about it, whether there was a drought or rainy season, or there were an insect plague, or whether there was a fire, just by looking at the cross section of this tree. So that's what I said. You can count the tree rings, and you can see what age the tree is. Now each tree ring marks a line between the dark late wood and then the early wood. If we just go back to this diagram here, we said that each year it marks the thing between the dark early wood. Here is your early wood, and here is your late wood. You can also see the early wood and the late wood nicely here in this diagram. This is the darker early wood there, and this is the lighter late wood. So this is in total is a tree ring. When we talk about a tree ring, it's not just the little dark bit there, it is this complete bit, yeah. The growth occurs in the cambium, remember where that is, and this is the thin continuous sheath of cells between the bark and the wood. Remember the cambium linked up with the parenchyma cells, which became meristematic, and then cells started dividing to the outside the phloem, to the inside the xylem, and this is where the growth occurs of the tree. Now this creates new tissues, and it increases the diameter of the tree in two places. On the outside of the cambium, where we said that the outer cells becomes part of the phloem. This is your secondary phloem, and then on the inside of the cambium, where it becomes part of the xylem, and this is the secondary xylem. Again, the function of phloem is it carries food that is produced in the leaves, and it carries it down to the roots. And then on the inside of the cambium, the xylem, it is where that carries the water and nutrients from the roots to the leaves. It's very important that you remember these functions. Now the xylem carries, okay, the cells show, it is these cells when you go and look at a tree. It is the lighter cells, it is these cells that shows the most variation, because here you will be able to see, is it broader, is it narrower, was there a felt fire, or was it a drought or not. So when the tree grows quickly, the xylem cells are large and they have thin walls, so it is lighter, And then when it grows slower, the xylem isn't as big and it has thicker walls, so it is darker. So basically, if we look here, it is where it shows the most variation. If it is the early wood and the late wood. Earlier on, the tree grows faster, so the xylem is bigger. The cell walls aren't as thick, so it is lighter. And now when it is later in the season, it grows slower. The xylem's walls are not as thick. It is thicker, so it is darker. Now, what causes variations in the tree rings? I've mentioned a couple of conditions, so we can look at this a bit more in detail. So, encouraging conditions. This is when it is optimal for the tree to grow. When there's enough water and enough nutrients, then the tree adds extra tissues and it produces a thick ring. When conditions aren't optimal, the tree doesn't add as much tissue and it doesn't have as thick a ring. Now, what causes these variations? If you have a higher springtime temperature, so that means when it's a longer season, spring starts earlier, then it will have a wider ring because there's longer time for the tree to grow. If there's a lower springtime temperature, in other words, spring starts later, there's not as much time for the tree to grow, it will be a narrower ring. If there's a lot of rainfall, then it produces a wider ring because more water means there's more chance to grow, it's better growth conditions, so it will be a wider ring as well. More tissues are created, so it will be a wider ring. Now, you must remember that different species of trees might respond differently to the weather conditions. One might respond strongly to rainfall changes and the other one not so much. So you would not, yes, you would have to take this into account, how a tree would, if you want to see what the temperature or the weather condition was over a year, you would have to take into account that different trees respond to weather conditions in different ways. Some are not as affected as others. Now, crowding from neighboring trees. That means when it's a forest and it's a very thick forest and there isn't much room for the tree to grow, you can think that there's not much room for the stem to expand, so the rings will be narrower. And then when suddenly, if you see that the rings were very narrow and then all of a sudden they start broadening, you can assume from that the tree was crowded, and then the trees that was crowding it was removed, and then suddenly it could grow. If you have a situation where the rings are close to each other on the one side, and then on the other side they're further apart, you can see that the tree was only crowded from the one side. So on the one side of the tree, there was a tree and it couldn't really grow on that side, but on the other side it was fine. This is what I said, many narrow rings, and it means, yes, and then suddenly they're removed, and then fire scars, of course, suggest that they are past a forest fire. So you can go back and you can see on this ring, let's go back to this diagram again. If we go and look at this here, we can go and count the rings and we can see how old the tree is. And then we can go and count and we can see, okay, well, this was a very good year. It was very broad, so there was lots of rainfall here. And then here you can see, okay, so when the tree was this old, there was a forest fire. And also here you can see, yes. And then this was a very droughtful year. It wasn't lots of water, so the tree didn't grow as much. And then also you will get scars that's due to insect plagues, and you can see that year there was a plague of insects. Well, that wraps it up for our stems of dicot trees. We're going to quickly take an ad break again, and then we're going to start with some questions, typical exam questions that you can use to prepare for. Welcome back from your break. I hope you enjoyed it. Right before we went for the ad break, we stopped and looked at the internal and external structure of dicot stems. Now, we have looked at the dicot stems, we've looked at the dicot roots, and what we're going to be doing now is look at some typical exam questions you can expect. Now, the first one we're going to be looking at is this one. Which of these types of cells are most likely to divide? Now, we have the option of parenchyma cells, meristem cells, epidermis, and the xylem. Now, for you to be effective in answering these questions, you need to know the function the function of each of these cells, and we're going to be looking at that. Now, first of all, you remember your parenchyma cells. You can remember when we looked at the parenchyma cells, these were the most abundant, and they were the big cells, and they were used for storing water and minerals that were absorbed. Now, your meristem in your root, this is a cells. Basically, I'm going to be giving you the answer now. The meristematic cells is where cells continually divide by mitosis. So this would then be typically your answer. These are the cells that are most likely to divide. But let's just look at the other ones as well. Now, your epidermis, this is the cells right on the outside of your stem and of your root, and this is a single layer of cells, and they are there to protect all the tissue inside the stem or the root. And then you get your xylem, and your xylem was found, it is basically the tissue that is responsible for taking the water that is absorbed from the roots, and it takes it to the rest of the plant. So that would be then your answer here would be the cells that are most typically to divide would be your meristematic cells. Just to refresh you, if we go and look at your diagram of the root typically, remember you had your root cap right at the bottom there, and this was to protect your root, and it's also covered in a mucus slimy substance, so it makes it easier for your root to penetrate into the soil. Then behind that you got your meristematic region. This is now in your root, and this is here that the cells will continually divide by mitosis to elongate your root. So it's the meristematic cells that will divide. Our second question, which of these cells in a plant is apt to be non-living? So which cells, if you look at these different options that you have, the parenchyma, the collenchyma, the sclerenchyma, and the epidermal cells, which of these is most likely to be non-living? Now again, you're going to have to refer back to your cell structures and everything you learned about the different types of cells that you get to fall back on this. So let's go look. Again, you have your epidermal cells. Your epidermal cells are right on the outside, and this is to protect the inside, and also root hairs form from your epidermal cells. So again, the moment I think that root hairs can form from epidermal cells, we know it's living. Then your parenchyma cells, these cells are where you store your water and minerals again. The collenchyma cells, it's also for structural support, but they contain the chloroform. They're more on the outside, specifically on the leaf, and this is where you get your photosynthesis from, and then you get your sclerenchyma cells. And these are the cells that are mainly dead, but they are very strong cell structures, very strong cell walls, so they are there for structural support. So the answer here would be C, which is your sclerenchyma cells, and they are non-living. All the others still have a living function. Now question 1.3, root hairs. In which zone will you most likely find the root hairs? So let's look at your structure of your root again. You have your root cap, and then behind your root cap you have your meristematic region, then you have your region of elongation, then your root hairs are found behind there, the root hair region, and then you have your more mature region here where cells grow bigger. So this is the area where you would find your root hairs. So it will not likely be in the area of cell division, the meristematic region. It will not likely be in the area of elongation, in the area of maturation. This is here at the top. Remember where I said where they grow bigger, and this is where they grow more mature. So basically, if we go look here, your meristematic is here between your meristematic region and your root hair region. That is where you would find your root hairs. So if we move on to question 1.4, where is the cortex found? Do you find it in the roots, stems, and leaves? Do you find it in the roots and the stems, in the roots only, and in the stems only. Now let's quickly go look at the structures of the root, stems and leaves and see if we can find this answer. So this is your root. Let's quickly look at the structure of your roots. You get your epidermis on the outside, then you have your parenchyma cells, and there's your cortex. So yes, definitely you do get the cortex in a root. Let's just look at our... So yes, the root would definitely be one. There it is. Then you get your endodermis, your endodermis, your pericycle, your xylem and phloem, which all makes up your stele. So that is your root. So we see there is a cortex in the root. Here you have your stem. Again, you have your cuticle, the outside layer, your epidermis, your Casparian strips, your endodermis, the pericycle, your phloem, your cambium, your xylem, the xylem parenchyma. And again, you have your parenchyma cells, and yes, we do get a cortex in the stem as well. So we can definitely say we have a cortex in the stem as well. If we go and look at the leaves, the anatomy of a leaf, which we didn't touch on this lesson, but just for quick revision, this is what a cross-section of your leaf would look like. You also get your xylem and phloem. You have your lower epidermis, your upper epidermis, the cuticle, which is very important in the leaves because they have a very big surface area and it prevents water loss again, and the vein there. So in the mesophyll cells, the palisade mesophyll cells. So again, there's no, in this one in your leaf, you can see there's no cortex. So the correct answer here would be, it would be the roots and the stems. So the answer is the cortex is found in the roots and the stems. Just a note here that you can see how important it is for you to know your diagrams and where the different tissues are situated in your plants. It helps you a lot because the minute you know that, you can work from that. So it's very important for you to know your diagrams and how it looks, your internal structure of your stem and of your root. Let's move on to the next question. Between the bark and the wood, in the woody stem, there is a layer of meristem called what? So we're looking between the bark and the wood. So immediately we're talking about a stem. So we're going to be looking at a structure of our stem again. So let's start from the outside. You have your periderm, you have your bark right there. There's your bark. Then you have your secondary phloem, your vascular cambium, where remember where growth happens in this direction and in that direction. From your 3 circles, your xylem, and which is your secondary xylem, and then your secondary phloem. So we want to know that between the bark of the wood and the woody stem, so here is the bark of your wood. There, this whole area is the bark of your wood. And the woody stem. Just there. What do you find? And the answer would be your cambium. So it would be your vascular cambium. So let's move on now to question 1.6, which has to do with the annual rings in a tree. Now the question is, the annual rings are the number of, your first option is internodes in a stem. Now if you go and think back to your external structure of a stem, your internodes was this area. So you had your node there and a node there, and it's this area here that we call the internode. So no, it is not the internodes of a stem. The rings are vascular bundles in a dicot stem. Is it the xylem and the phloem and everything? Let's go, what was the vascular bundles made of? If we go back to that one, the vascular bundles in a dicot stem, we can go. the vascular bundles, which is your phloem and your xylem and your parenchyma and the cambium. So it is not completely that either. The layers of secondary xylem in the stem, we go and look let's first go and revise what was a year ring or a tree ring made up of. You have your light area and your dark area, and it is basically both the dark ring and the light ring that makes up your ring. So if we have to look at this, so it would basically your light area and your dark area, and we would go back and it is your secondary xylem in your stem. The correct answer would be C, because you first get your lighter area and then your darker area. And this is what would make up your tree ring. The Casparian strip is found where? Between all epidermal cells, between your xylem and your phloem, on four sides of your endodermal cells, or within the secondary wall of the parenchyma cells. And let's just remember, if we go back, and I think a good example here would be the diagram of your root. We said that your Casparian strip is the band around the endodermis, and this is to make the movement of water into your xylem possible. So you would find your Casparian strips right there. If you look at it more detailed, this is where you would find your Casparian strips. Your endodermis and the Casparian strips would be there. So if we look at our options, the right option would be on all four sides of your endodermal cells. Now question 2. This question says, study the photomicrograph which shows several important tissues in a typical stem. So we are given the stem, and then the question is, the first one it says, is this a stem of a dicot or a monocot stem? So it is important for you to know, in this situation, this is a dicot stem because it's got the typical structure of the dicot stem. You have your epidermis, you have your, basically the thing that gives it away is if you look at your vascular bundles, and they are situated in a circle around the stem. Is that diagram that I drew for you here, where I said that this is your stem and the vascular bundles would be situated in a circle in the stem, like that. Okay, the answer to this question is it is a dicot stem. Now let's move on to question 2.2, where you use the same photomicrograph and you now have to provide labels for A to D. So A there, it is right on the end, the right outer layer of cells of your stem. So A would be your epidermis. We can move up. A is your epidermis. B is there, and if you look, this is one of your vascular bundles, and these are the cells there, so this would be your phloem. C there is still in your vascular bundle, it is that area there, and this would be your xylem. And then the last one, D there, you have your, it's on the inside. There it is your very large cells, and there's a lot of them, so you should know this would be your parenchyma cells. So if we just recap, you have A is your epidermal cells right on the outside. B is your phloem, it's in your vascular bundle there. C, it's your xylem, and then D is your parenchyma cells. So the next question, still on the photomicrograph, is to make a line drawing of a root. And when you make the line drawing, you have to use the following terms: your endodermis, your phloem, your xylem, your cortex, and your epidermis. Now the diagram I showed you in the beginning, this is typically the diagram I would want you to draw. So you would have your epidermis there, your cortex there, your phloem, your xylem, your Casparian strip, and your endodermis. So this is the cross-section or the line drawing that I would want you to make when we ask you this question. Now don't get confused with this photomicrograph. The photomicrograph is of a stem. The question here that they want you to make is of a root. So you would do your root. So that would be the one where you have your epidermis, you'd have your endodermis, your xylem, that's in the form of a cross, and your phloem, and this is your cortex. Yes, so you have your epidermis, is here on the outside. You have your cortex, you have your endodermis, your xylem, this is your phloem. So there we have our line drawing of a root. Next question, still based on the micrograph, is they want you to make a line drawing of a woody stem using the terms annual ring, bark, cork, phloem, pith, vascular bundle, and xylem. So let's refer to this diagram of us. And this is typically the diagram that you would draw. So if you look into this one, they ask you to make a line diagram, and now you have to use the following annotations. The annual ring, the bark, here you find the bark. So that whole area there, that is the bark. Your cork, here is your cork. This is the area on the outside, right on the outside, even of your tree. Your phloem, here is your phloem. Right there is your phloem, your cork, we have your bark. Then you want your pith. Your pith is right there in the middle. This would be your pith. There, your vascular bundle. Your vascular bundle, remember, it's made up of your phloem, your xylem. So that would be here would be your vascular bundle. So you would basically do this. And if we can just add this drawing here, your vascular bundle would be there. So your vascular bundle. And this is a section taken from here, like that. Well, that's what I said. So this slice here is what you see there. So you could just make a little drawing there and refer that this is this area there. So we have our annual ring, our bark, our cork, our phloem, our pith, our vascular bundle, and our xylem. And then our annual ring, the growth is in that direction and here. So it is our xylem. It's from here. Right, so that is typically what they would want you to do when they ask you this question. We can go on to the next question, the last question we have. Now question 3 is a true-false kind of question. So what they say is change the false statements to make them true. So you now have to go through each of these questions and see what is wrong with them. So question 3.1. The growth points of a stem is called the anthers. First of all, we have to go through these questions and see which is true and which is false, so that we don't change the one that's wrong right. So the growth points of a stem is called the anthers. This is wrong. It's false, because the growth points of a stem is called the meristem. It's not called the anthers. This is wrong. 3.2. Parenchyma cells are the most abundant cells in most plants. Is this true or false? We know that this is true. So there's nothing to change in this question. It stays as it is because it's true. 3.3. Vascular tissues are clustered at the center of a root. We know that this is true. If you just recall back on your vascular tissues and your root, you remember that when this was your root, the center of your root, and in the center, in your endodermis, you had your xylem and your phloem. So it is in the center of the root, so this is true. Nothing needs to change with this question. Then wood consists mainly of xylem. We just discussed that in the previous question where we said what wood is and the late wood and the early wood, so we know this is true. You don't need to change anything in that question. 3.5, the root cap absorbs minerals and water from the soil. Is this true or false? It is false. Remember, the root cap is the little cap right on the end of the root that's slimy and it's covered, slimy, so it protects the root and it helps with penetration into the soil. So this is false. It is, the root cap doesn't absorb minerals and water from the soil. The root cap actually protects the tip. It protects the tip. So with that, we've come to the end of this section on support and transport systems in plants. I hope this helped you a lot and that you've learned so much. Good luck for your exams, study hard. See you next time.